Excellent. Well, it's a pleasure to be on here and talk to you all. And I'll be really interested to hear more about the consortium and everything that you all are doing in South America too. So I appreciate the opportunity to present here, and also hoping to learn from you all as well. So I'm going to talk about freshwater mussels and the ecosystem functions they provide, and some of the conservation opportunities that I think we have here, and then also globally, and how we can think about conservation of this taxonomic group. But as I get going, I just want to acknowledge the fact that the work that I'm presenting is not just my own. It's come from the work of many students, collaborators, and folks that I've been working with over the last several years. So I certainly want to acknowledge postdocs, grad students that I work with, and several collaborators and undergrads that work in my lab as well. So without that, a lot of this work that I'm about to talk about wouldn't be possible. And then on top of that, I also want to just put in a lot of the funding sources, especially the National Science Foundation. So when we look at this riverscape, right? So you're looking out on to the river, and when I'm conceptualizing it and thinking about how these streams, how rivers function, one thing I'm thinking about is what are the inputs into this river? So we can think about the nutrients that are coming in, like dissolved organic carbon, nitrogen, and phosphorus. And then also overlaid onto this riverscape, we think about the aquatic organisms that live within these systems. So, everything from our freshwater mussels to the trees, the riparian cover along the riverbanks, the fishes, the aquatic insects, and whatnot. And these species impact how these rivers function. However, ecosystems are changing globally from a multitude of reasons. So everything from changing inputs into these ecosystems, oftentimes enhanced runoff in the form of nitrogen and phosphorus. Also due to global climate change, which is, a really large problem globally. And then I know a lot of folks in the Andes and the Amazon are thinking a lot about this, changing temperatures and also the hydrology of these systems. So collectively, all of this has impacts on the species composition and resulting in loss of biodiversity. And we know that freshwaters are experiencing declines in biodiversity that are much greater than those in terrestrial or marine ecosystems, for that matter, with extinction rates being as high as 4% per decade. And as you can see from this figure here, mollusks are at the top of this list as far as the decline and what's imperiled. But when we're thinking about this and how the decline of these species are, what is the actual impact to ecosystem structure and function? Because there's really little that's known. And so there's this need to make better and more mechanistic hypotheses to understand how loss in biodiversity links to ecosystem function. And so one of the approaches that I've been taking and others have been taking in other groups like plants and whatnot is linking these multiple scales of biodiversity. So we can look at this phylogenetic tree and understand how species are related to one another. And oftentimes traits of species are phylogenetically conserved. So we can think about traits such as body size, their fecundity, morphological traits, etc. And so these functional traits, from their morphological, physiological, structural traits, they impact fitness and how the species performs in nature. But not all traits are phylogenetically conserved, but generally, given this phylogenetic conservatism, it can allow us to make some predictions. And then when we're looking at the community, these traits are filtered, and we see the community structure of that. And these species traits all affect what's going on in the ecosystem. And so we can think about these species effect traits, such as their nutrient cycling, i.e. in their stoichiometry, or like their grazing ability, those functional effects, and understand how they're actually affecting the ecosystem. And so this encapsulates the diversity of ecosystem processes that species or a community is performing in nature. And I will also mention that these traits can also vary intra-specifically, translating into differential ecosystem impacts. But by linking these multiple scales of biodiversity, we can better understand how we can better make hypotheses regarding how like a group of species affect ecosystem processes. And so you all are here because many of us work with freshwater mussels. So as you can see from this slide, we have a diversity of morphological traits. These are a bunch of species that I've worked with. And so I think they are a really interesting group for us to ask these kind of questions. And this, like I mentioned before, they're highly imperiled. So you can see here when we're looking across North America, South America, globally, many of these species are highly threatened. And then also have to mention, there's a lot of data deficiencies. There's so much more that we need to learn about this group. And here in where I'm working, so North America, we have a biodiversity hotspot. There's over 300 species in North America. And when we zoom in to the southeastern US, where I'm at, we see that when we're looking across fishes, crayfishes, and mussels, it's very high species richness. So you can see here that across all these taxa, there's a really high species richness. And then fishes, freshwater mussels, the Tennessee River Basin up here in North Alabama, it's very rich, but overall, we also have really high richness in the Mobile Basin. So in Alabama, we have 183 species of freshwater mussels. So for me, it's a really awesome place to work. And I probably don't need to tell this group, but it's a really diverse group. Many species have long generation times. Oftentimes, many species are long-lived, living from 6 to 100 years. And we see them, especially here, as they occur in these dense multi-species aggregations, at times like over 20 species, and these really dense aggregations of over, anywhere from 20 to over 100 individuals per meter squared. And as we know, generally as adults, they're fairly sedentary and they can't move very far. And they depend upon a host fish for dispersal, in which they have to get their glaucidia or their larvae upon a host fish to transport their juveniles in order to be able to be successful. And different mussels use different host fish. So overall, they're a really interesting group to study because of that. And then they have these really important impacts on ecosystem functions, as they provide biofiltration through filtering the water column. they are also important for nutrient recycling and storage. So as they're filtering, they're excreting nutrients, bioavailable nutrients back into the water column as ammonia and soluble reactive phosphorus. And they also excrete carbon that's highly available. And then also storing nutrients in their shell and their soft tissue. They also provide this really important structural habitat in the benthic environment, oftentimes being a really important component for other aquatic organisms. So you can see this caddisfly here that built its case on the freshwater mussel. They modify the substrate through their bearing, which I'll get into later, which is really important for biogeochemical reactions and the bacterial community. And furthermore, they're also food for other organisms such as turtles and fish. So within this group, we see high interspecific diversity. And, there's, currently taxonomic revisions. I'm always like trying to figure out what species I'm working with because the names seems to be changing. But within the subfamilies of Unionidae in the subfamily Amblyminae, we see a really high diversity of taxa. And so we can use this phylogenetic information for predictive biology, right? So by understanding how these different species are related to one another, we can start to put together what kind of functions these different taxa play and use this for predictive biology, and also maybe understand something about their like how susceptible they are to stressors in the environment as well. So I'm going to talk about the area I work in, the Mobile and Tennessee River basins. And so within these river basins, this encapsulates about 60% of mussel diversity in North America. So it's a really awesome place to work. So here you can see in the blue, this is the Duck River in the Tennessee Basin, and here's the Sipsey River on your right in the Mobile Basin. And so I'm going to talk about species traits, and is trait variation in mussels predicted by phylogenetic differentiation? And so the traits I'm going to focus on today are body stoichiometry, their feeding traits, so what are they eating? And I'm going to also just briefly cover a trait data set that we published last year. Some of the people on the call, I believe, were involved in this. If I had time, I would touch on morphology, but I will say it's not really necessarily predicted by phylogenetic differentiation, and I'm happy to talk about some of our morphology data later on, but these are the topics I'm going to cover right now. So just to get you all kind of situated on stoichiometry and how stoichiometry impacts ecosystem function. So generally, animals are considered to be stoichiometrically homeostatic. So we maintain a constant carbon to nitrogen to phosphorus ratio. And so if you were, for example, to eat spinach every day, right? We have like a lot of proteins and whatnot that have a lot of nitrogen. So we would try to keep the nitrogen that's in that and get rid of everything else in excess. So get rid of the excess carbon, for example. And so animals do that. So as they're feeding on different resources that have various carbon, nitrogen, and phosphorus ratio, as they're consuming these resources, for example, here in this example, they're maintaining more phosphorus in relation to the resources. And so they'll excrete excess nitrogen in relation to phosphorus. And so the excretion and ingestion of nutrients are proportional to that ingestion of the element, and then also what that animal requires. And, these rates scale with body mass and temperature. And so this is a way in which animals are affected by the environment, but also how they're impacting nutrient recycling in the ecosystem. And these rates and stoichiometry varies across different taxonomic groups. And so this allows us to understand how an organism, what its needs are, and then also how it's affecting ecosystem function. And so just to jump into some data really quick, here's nutrient stoichiometry within a single river system. This is in the Sipsey River. And what you'll see here, these bubbles on this plot are all these different taxa. And you can see that they're kind of separated in this three-dimensional space. Furthermore, we see that when we're looking at their volumes of their pretty much their three-dimensional stoichiometry, that certain taxa have a larger volume, in which the taxa that are in this lampsaline tribe within that subfamily Amblyminae have a larger niche volume than quadrilinees and pluribemenes. And then when we're trying to examine if these taxa, if this is actually phylogenetically conserved, we'll see that as we have greater phylogenetic dissimilarity, that they are more dissimilar in their tissue stoichiometry space as well. So suggesting that indeed, as these organisms are more dissimilar in that phylogenetic space, that they are also more different in their tissue stoichiometry, which then has an impact on how they're functioning in the ecosystem. So when they're excreting nitrogen back out into the system, which is highly bioavailable and really impacts the algal community and the biofilm community, we see that taxa that have a higher nitrogen to phosphorus ratio excrete a lower nitrogen to phosphorus ratio, right? So this makes sense because if they have a higher requirement for nitrogen, they're going to excrete less nitrogen, right? I know it's hard to think about ratios, but this overall meets the expectations of ecological stoichiometry. So given this phylogenetic signal, this allows us to make predictions on how these taxa overall impact ecosystem function. And then furthermore, when we look at like how this is related to some of their life history traits, right? When we look at their growth rates, so as species have a higher growth rate, they have a higher soft tissue percent phosphorus. And this makes sense based on what's called the growth rate hypothesis, as phosphorus is required for higher growth rates due to the allocation to RNA. And so we see that as they have this higher growth rate, the soft tissue has a higher percent phosphorus. But interestingly, we did a follow-up study on their shell tissue, and animals that have a higher growth rate tend to have shell tissue that has a lower percent phosphorus, or a lower amount of phosphorus. And many of these species as well with the higher growth rates have thinner shells, and they're more fast growing, so they're allocating less phosphorus in that regard. All right, so I'm going to switch over to mussel feeding. So a lot of the work I'm about to present now is from the work of a PhD student in my lab. This paper is currently in review. And so Matt, he looked at the cirri density. So essentially the cirri, you can see here in the gill, and we can see that the cirri density varies across different species. But also when we look across different sites, there's a lot of variation, right? So you look here, for example, quadrula varicosa, the pistol grip muscle. You can see here at one of our sites, it has, pretty high series density, and then you go to the site 2, and it's quite different. So, this density of the series translates in the size of particles that they're able to filter, but we see that, yes, there's differences between species, but it seems to be pretty phenotypically plastic across sites. And so we paired these data with stable isotopes. So stable isotopes can give you an idea of what food resources organisms are actually assimilating into their body tissue. And I know this plot is a lot to look at, but one thing I just want you to note from looking at these data is that you see that species are generally different in this isotopic niche space. There is some overlap. For example, look at site 5 and 6, there's, a good deal of overlap, but also there's variation. And I think that's super interesting because we all categorize mussels as filter feeding bivalves. But when we look at what they're actually assimilating into their body tissue, they're doing something differently. And so if we combine our stable isotope data with our series density data that we generated, we see that as we have a higher series density in these mussels, that these standard ellipse areas, so the size of these circles, declines. And so as mussels have a higher series density, they occupy a smaller trophic niche. And then when we're going back to the phylogenetic stuff I was discussing before, so if we standardize our isotope data across all of our sites and look at the relationship between their isotopic, their kind of like mean of the mussels, we can see that as they are more phylogenetically dissimilar on this x-axis, their distance in this isotopic space is more distant. And so this suggests that species that are more related to one another are more apt to feed on the same food resources. All right, and so last on the trait stuff, I just wanted to put this out there. I think there's a few folks on this call that were part of this, and I think this could be an opportunity also for South American researchers. I'm talking to some European colleagues right now to try to put this together for the European taxa, putting together the traits that we know about mussels. And so this is for the mussels in the US. And so we put this out just last year. And so this is information on their distribution, body size, habitat, conservation status, and just what available genetic information is out there. And, it puts together a lot of information on these taxa, but I think the really interesting thing that came out of this was also identifying the data deficiencies we have. So if you look at this figure on the right, so this is grouped by those phylogenetic tribes, right? But what you can see here is that, yeah, we know how big these animals are, right? So we have max length. However, when we look at several other traits, such as their fecundity or even longevity or maturation age, there's very little known. And so I think this really highlights some research directions that we can go towards to better understand this group. And yeah, European and South American mussels. And I think like linking these traits could really help us also understand how things are going to change in relation to things like climate change or nutrient pollution, right? And how that's going to impact ecosystem function. So for example, if we're thinking about an organism's vulnerability, let's say to temperature, if traits like, let's say, whether it's like their feeding or their nutrient, like how they're excreting nutrients to the environment, if those traits are distributed like this and we lose all the animals on this high vulnerability side, you know, there might not be many impacts to ecosystem function in relation to if vulnerability and traits are distributed in this kind of way. That could have a very differential impact to our river and other freshwater ecosystems. So I think this is really essential to determine for future management strategies. All right, so I'm gonna turn a page here and talk about freshwater mussels' impacts to ecosystem function. So, you know, hopefully I've convinced you that even though mussels are all filter feeding bivalves, they're doing things a little bit differently. So they're excreting nutrients a little bit differently. They're feeding, or like at least assimilating food resources differentially. And there's other things that I could talk about, but so now I'm going to talk about how do mussels impact function, and does species composition actually matter when we scale up to the ecosystem? So I'm going to go into these functional effects. So like I talked about how they have different nutrient stoichiometries and how that impacts nutrient availability. So nutrient excretion by mussels can actually exceed background nutrient conditions and demand. So in these rivers where they're incredibly dense, occur in these dense aggregations, they can be a major source of nitrogen, phosphorus, and labile dissolved organic matter. So what you're seeing in this plot here, so this is volumetric excretion. So it's scaling what they're excreting to the volume of the river and background nutrient concentrations. And what you'll see here is that mussels, so this effect size here, so if it's above, if it's in the positive zone, that means it meets or exceeds background nutrient concentrations or conditions. And what we see here is that mussels can meet approximately 30% of the demand of dissolved organic matter. Not super important for dissolved organic carbon, but you know, about 2.5%. But for nitrogen, they certainly are really important. This is during base flow conditions where they can meet and exceed background nitrogen demand, and can also meet up to about 20% of the phosphorus demand of the ecosystem. And so this is really important in fueling the food web in regards to algae and heterotrophic microbes. And so, you know, the loss of mussels could really change these ecosystems, and certainly has. And we can also look at this at the reach level. So if we zoom in at this river reach scale, we can see that these organisms can act as these biogeochemical hotspots where they can, when we're mapping out their distribution. So each of these points that you see on this map is a quadrat, a quarter-meter squared quadrat that we dug. So we know the species composition. We've measured their nutrient excretion rates, and so we can use these data to actually scale how they're affecting ammonia availability, phosphorus availability, and then here this is looking at their biodeposits, so essentially their adjusta and what they're putting into the benthic compartment. And this nutrient excretion and their relationship, or how they live at this oxic-anoxic boundary layer, also has an impact on microbially mediated denitrification. So denitrification is the permanent removal of nitrogen from a freshwater ecosystem in the form of N2 gas. But this is a microbially mediated process that occurs at the oxic-anoxic boundary layer in stream or other freshwater sediments. And mussels can play a really important role as they bury into the sediment. They're excreting bioavailable ammonium, and that can be converted into nitrate. Furthermore, they're biodepositing materials into the substrate, which is really microbially active, provides a really important energy source for these denitrifying bacteria. And so in an observational study in which we were essentially digging our quadrats right before we dug our quadrats for our mussels, we took a sediment core. And what you'll see here is that as we have areas, so just essentially quarter-meter squared quadrats with higher biomass of freshwater mussels, we have higher denitrification rates. So suggesting that, you know, these animals are strongly impacting this microbially mediated pathway of nitrogen removal from the ecosystem. And then furthermore, if we look at just the quadrats in which there were mussels there, the species trait of the mussels that are in those quadrats play a role. So if we scale the excretion by what taxa are there, species play a really important role. So as we have taxa that excrete at a higher nitrogen to phosphorus ratio, this is explaining the residual variation in this plot with denitrification. And we zoom in and look at the correlation with certain taxa. So lampsolines, they tend to have a high excretion nitrogen to phosphorus ratio. They're positively correlated with higher denitrification rates here, right? So they have a higher biodeposit nitrogen to phosphorus ratio, and these species are playing a larger role in regards to positively impacting denitrification versus other taxa that have a lower excretion nitrogen to phosphorus ratio, are more negatively correlated to denitrification. So this really highlights the fact that not only are they having a direct impact on nitrogen and phosphorus availability, that has a consequential impact on microbially mediated nutrient pathway of denitrification. And so we can use these kind of data to scale up to the ecosystem level, right? So we can use these species traits. How far do they bury? What are they filtering? What's their nitrogen to phosphorus ratio that they're excreting into the ecosystem? What are they biodepositing? And then take it across different species and scale it up to understand what are they doing at this whole ecosystem level. And so I'm just going to give an example of how we can do this really quick. So for example, this is a site that we've been studying on the Sipsey River for 9 years now. So we've been sampling the particulates, so the ceston that's coming downstream, and during base flow conditions, we can estimate how many moles of carbon, nitrogen, and phosphorus are coming down the river. And then based on filtration data we've collected on different species, we can estimate that, you know, these animals in this one particular aggregation can filter approximately 16% of that particulate flux during base flow, so low water conditions. And furthermore, when we're looking at their biodeposits, so out of what they're filtering, what are they adjusting and putting or releasing as pseudofeces, approximately 20% of that filtered material is deposited in those, so providing that microbially active substrate for other activities. And then when we're looking at, if you want to look into this, Jonathan Lopez, we published a paper in Ecosphere earlier this year. So this is just looking at the production of biomass in the environment. So approximately 1% of what these animals are filtering actually ends up into muscle biomass, or them actually accumulating biomass. And so, and then they're kind of important for the dissolved flux. And then we can also look at this assemblage and look at how much denitrification that's being provided. So I think it's really interesting to think about how these organisms are a really important energy and nutrient pathway in these ecosystems. So the last thing I'm going to talk about is kind of has more of a conservation angle as opposed to an ecosystem function or ecosystem services angle. And so I'm going to talk about freshwater mussel diversity across levels. And so overall, does species and genetic differentiation translate into differences in functional diversity? So I've talked a lot about how these organisms have different traits and how those traits can manifest into differences in ecosystem function, and how mussels play a really important role in ecosystem function. But how does diversity across different levels translate into these functional effects? Going back. So yet again, this work was done in these 7 river basins and the Tennessee and Mobile River basins across 22 sites. And so first, I just want to highlight the fact that at least in these river basins, as we have higher mussel density, so sites that have higher mussel density also have higher species richness, which makes sense. So this is the more individuals hypothesis, essentially. So as you have more individuals, you're more apt to have higher species richness. Furthermore, as we have sites that have higher mussel densities, we also see a greater abundance of imperiled species. And generally, sites with higher species richness also host a larger number of imperiled species. So essentially what this is saying is, if you want to focus your conservation efforts and you want to maintain species richness, sites with higher mussel densities should be your target sites for conservation. Furthermore, if we're looking at genetic diversity, so this is some work that was done by a grad student, Jamie Buchholz. She's now a postdoc at Virginia Tech. So what we found here is that as we have sites with, so we did not find any support for higher species richness being related to genetic diversity, but we did find that sites where you have a higher density of that particular species, you got a greater genetic diversity. So what this is saying is that if you want to maintain genetic diversity in a specific species, or you want to identify a site with high genetic diversity, find sites that have a high abundance of that species. And so this could be really important when you're doing reintroduction efforts, for example. So these are all these species, these aren't listed species, but if you were trying to propagate a species and reintroduce it somewhere, for example, you would want to, the source population that you might want to target, you might want to target a source population that has the highest density to have the highest genetic diversity. And then lastly, if we're thinking about these functional effects, so this functional richness encapsulates their body size and those nutrient stoichiometry traits I was telling you about before. So if we want to have higher functional diversity and have a diversity of nutrient recycling, body size, etc., sites with higher species richness are going to be the most important for maintaining function and having high functional richness in that ecosystem. But we're still pretty unclear on how intraspecific variation will impact function, but generally we predict greater diversity enhances function. And then lastly, when we're looking at genetic diversity, we see, right, yet again, if we have higher species densities, we see generally at the site level, higher levels of genetic diversity. We don't see any patterns between species richness and genetic diversity, as I mentioned before. But we do see, to some degree, as you have higher mean genetic diversity at the site level, we see higher functional richness, but it's not a very strong relationship. So overall, greater density translates into higher diversity, but this greater genetic diversity doesn't necessarily mean higher levels of functional diversity as far as what traits are being expressed at that ecosystem. And I think this is important as we think about our conservation goals, right? So if we're thinking about our ecosystem services, right, we might have like different strategies in which we try to maintain these ecosystem services. So if we're thinking about like Clean Water Act, we want to maintain these sites that have the highest density. And so sites with the highest density and abundance of mussels is going to be the most important for maintaining clean water, et cetera. Versus when we're thinking about freshwater biodiversity conservation, when we're thinking about the Endangered Species Act and the US, we're always targeting rare species. And those might not always be the most important for ecosystem function. But, in regards to the strategies, some of those strategies are quite similar. But what I'm trying to get at with this relationship between density and species richness and the genetic diversity is that a lot of these conservation strategies can come hand in hand. So a few take-homes from today is that despite the fact that freshwater mussels are all in the same functional feeding group, they're a really interesting and important model taxa as we can make predictions regarding how they're related, and they're doing different things. They're feeding differently, they're storing nutrients differently, they're recycling nutrients differently that have impacts on ecosystem function. And then from the trait paper that I briefly mentioned, we do need more information on species traits, and then we also need more population level data. We could very much refine that data set. And then lastly, more diversity across scales translates into diversity and function. So as we have greater diversity at the species and intraspecific level. We have, more diversity and ecological function that these communities are providing. And so just to leave it on some things that I'm really interested in right now, some current and future directions. We certainly need to learn more about the host-fish relationships. As you can see here, most of the data we have are on lab transformations and lab work, essentially, and not a lot of data from the actual field. And so there's a lot of work that could be done on these host-fish relationships. I'm also interested in mussel impacts on the physical environment and some of the work. We're following up some work on some of the denitrification work. And then we also need to better link what species are vulnerable to things such as temperature, climate change, to understand how communities might change in the future, what that means for ecological function in our freshwater ecosystems. And so with that, I really appreciate your attention, and I'm happy to take any questions.